My name is Tyler Jacks. I'm the director of the Koch Institute for Integrative Cancer Research at MIT and a professor of biology at MIT. And I've been at MIT since 1988. I was a postdoctoral fellow with Bob Weinberg in the Whitehead Institute at that time. And then I joined the faculty in 1992, and I've been here ever since. Prior to coming to MIT, I did my PhD work at the University of California, San Francisco with Harold Varmus, who is currently the director of the National Cancer Institute, and I was a college student at Harvard. I've been doing cancer research for a long time, actually. I started as a college student. Summer after my sophomore year, I started working in a laboratory that was doing molecular genetic research related to cancer, and that's really what piqued my interest in the field, and went off to graduate school to study cancer research and continued on from there. My interests have largely centered on understanding the genes that get affected as cancers develop from normal cells. And my laboratory now works on several of those genes, trying to understand how, when they become abnormal, when they function improperly, cancer cells arise. Well, my laboratory is involved in cancer genetics and specifically in developing mouse models of human cancer. We have a major program in lung cancer, but we also work on pancreas cancer, colon cancer, ovarian cancer. We've worked on brain tumors in the past. So we use genetic engineering in the mouse to create novel strains of mice that are cancer prone. And we use those mice to understand the biology of cancer and how it develops and progresses, and increasingly how we can reverse that process with various forms of interventions, small molecule interventions and immunological interventions, RNAi-based interventions. And these models that we and others have developed have turned out to be a very useful tool, again, not just for understanding the disease, but understanding how to better control it. And speaking of industry, many of our models have been licensed to companies to test their drugs in more appropriate or more predictive animal models. And so we think that this is an important tool in teaching us what's the best drug for this specific form of the disease, in a way that gives us information that allows fast tracking of those drug testing exercises in patients. Right now, clinical testing is a very long, very expensive, and very uncertain business. If we can de-risk that process by giving better information in the preclinical setting, it should make the process go faster, more efficiently, and also less expensively, which I think is a very important thing.